So basically, we're going to talk about some things, and we are going to diagram some of them on the board for emphasis, but I just want you to sort of appreciate that you pretty much know everything you need to know to understand everything in this chapter, minus the terminology, which is kind of annoying, but we'll just get through it. Now, just to talk about some of the practical applications of immunology, obviously one huge thing that has been, a revolution in medicine is vaccination. And we've talked about how vaccination started. It was actually a practice more ancient than your textbook suggests to inoculate people with smallpox to prevent them from getting sicker later on. Now, this came with some risk because you were inoculating them with real, actual virus. And so about 1% of the time, this was not such a great thing for the inoculated person. But given that smallpox at the time had a 50% lethality, that was kind of a fine risk to take. Subsequently, different people have come up with versions of vaccination against smallpox that have a much higher safety record. For instance, you can inoculate people with cowpox virus. And does anyone know the virus that's currently used to inoculate people against smallpox? Vaccinate them. Vaccinia, right? Which is interesting because we actually don't know that much about vaccinia other than that it's very effective and it doesn't make you sick. Pasteur of pasteurization fame also was in on the vaccination game. He came up with a way to inoculate or to vaccinate against rabies, which was actually also really kind of a game changer. And obviously, although we don't use this now in human populations except for as a post-exposure prophylactic, we use it very frequently in domestic animals. So here's some terms that you should be able to help me to define. So when we're talking about exposure to something that your immune system would recognize, what is the primary response? Say again? It's your first exposure, right? Yes, you are attacked by the thing. And what is happening in that primary response, besides our innate immune system kicking in? What is happening in the adaptive immune response in that primary response? What? Yeah. Antibodies on what cell? On B cells. Also, what other cell types involved in finding the whatever it is? T cells are helping. What else? Phagocytes, right? Phagocytes are talking to T cells. T cells are talking to B cells. B cells are so there's a sort of a communication among and a coordination among a lot of different cell types. That, of course, produces, after the immune system kicks the butt of whatever it is, then, if we see it again, what's that? Secondary response. Now, why is the secondary response in general a more effective? Why do we have a toehold? Why do we have an advantage this time? We have a buildup of antibodies. Remember, antibodies have a short lifetime. What do we actually have a buildup of? Memory cells. What kind of memory cells? B cells and T cells, right? So they can respond to this, and usually fast enough that you really don't get sick that second time in a noticeable way, right? All right, so what is this term herd immunity? More generally, it means that enough people in the population are immune, and you can accomplish this artificially by vaccinating people. Any idea roughly what percentage of the population needs to be vaccinated or immune for this to be effective? It's over 90%. Okay? Over 90% of people have to be immune, potentially by having been vaccinated or whatever, in order to provide protection against the other members of the community. Because if you don't have a lot of hosts, then it's very difficult to spread the disease, right? Now, does that mean that one person who's not vaccinated is completely protected? Absolutely not. They can still acquire it some way, but they're probably not going to get it from their friends and neighbors very easily. Can you vaccinate against bacteria or just viruses? Yeah, you can absolutely vaccinate against a variety of things. Anything that's antigenic that you can convince your immune system to recognize. It happens to be more effective in general to vaccinate against viruses. There's sort of complicated reasons for that, but you certainly can vaccinate against bacteria. Does anyone have an idea or an example of one that a bacteria they've probably been vaccinated against? Tetanus? Anything else? What else is in that tetanus shot that you usually get? Diphtheria. Why is vaccination so useful if you're dealing with viruses specifically? If we have a bacterial infection, potentially what could we do then? Antibiotics. Can we use antibiotics against viruses? No, we cannot. Why are antibiotics not effective against viruses? Yes, so that's getting at it exactly. The virus, in order to live, what does it have to do? Has to get into the host cell. So if you're going to kill the virus, one way to do it would be to target the host cell, which is not so great for you. And most antibiotics are targeted against bacteria. Absolutely. So there are viruses against bacteria. That would be what? Bacteriophage. They're going to be harmless to us, right? We're not going to notice them. But, some people have considered treatments against bacterial infection by chasing them with bacteriophage that would infect the bacteria. Absolutely. Okay. So here is just an example of how effective vaccination can be. So if we're looking at around the time when the measles vaccine was being developed and then widely deployed, the vaccine was licensed just before 1965, and we went from, 500,000 reported cases to maybe 20,000 cases in a few years. That's a huge drop. And then over time, as just this became a standard thing to do, we end up dropping the numbers really significantly. What other famous virus has been tamed by vaccination, eliminated from the world? Polio is on its way. The first one? Smallpox. Now, I want you to think about why it is that smallpox would have been a good one. Think about, for instance, have we been able to say eliminate rabies worldwide? Why is that? Where is rabies normally found? It's in animals. Are we going to go out and vaccinate all the animals on planet Earth? I'm not saying that probably there isn't someone out there who's thought of doing that. But it is definitely more difficult than something like smallpox, because smallpox is transmitted how? From humans to humans. If you're dealing with a pathogen that is only in the human population, then that is at least one thing in your favor in terms of trying to control it. If it's reservoirs in the soil, like what kinds of bacteria do we know cause disease that have a soil reservoir? Anthrax, tetanus, botulism, right? All of those guys that make what? Endospores that can survive a long time, and they are out in the environment. It's very difficult to treat the whole Earth. But if you're just dealing with humans, then that is one way. And our measles, are they a human-to-human pathogen, or are they an animal-to-human? What are they? They're human-to-human. So this is something you're going to be more effective dealing with this kind of thing. Now, is vaccination the way that you can have the biggest impact on disease? Not necessarily, right? So don't forget that we have antibiotics, right, to target some bacterial infections, although, of course, we're now in an era where that's becoming less and less effective. And this, access to clean water, is probably one of the most important factors worldwide. So if we just, for instance, look at the case of typhoid fever, which is a bacterial infection caused by Salmonella typhi. When we look at the sort of number of cases in the late 1800s, and then we see the advent of water treatments, filtration, and then chlorination that were antimicrobial, remove microbes or kill them, we see a huge drop in typhoid fever. Obviously, something like whooping cough in your vaccinations, when you give vaccinations to kids, whooping cough cases go way down when you increase the vaccination. But it's important to recognize that is this kind of change. Okay, so in that previous graph, the scale was blown up to make us feel good about ourselves. Oh, look at what we did. But if we actually look at the scale in the human population worldwide, we're talking about, this kind of difference versus the kind of difference that knowing what the organism is and being able to understand how it's transmitted and make those kinds of changes. And some of the changes that would make the most impact worldwide, again, are very simple. Access to clean water, very simple thing. So polio, measles, tuberculosis, whooping cough are these things that are really a huge problem here in the United States. In the world, they're still a big problem. So we have to, for instance, measles, something that probably none of us think about as being particularly dangerous because we're all, pretty much come into the world and get the vaccination right away, still kills hundreds of kids every day in the world. So let's think about the types of vaccines that people make. You will need to know this. So attenuated whole agent vaccine, what is that? What is that going to mean? So this vaccine, attenuated whole agent, is this the original disease-causing organism? What's changed about it? Less concentration? Yeah, sure, that's part of it. Attenuated has a specific meaning. It usually means weakened in some way. So it could be, for instance, be a genetic variant that's a little bit less virulent, but still has the antigens that would provoke an immune response. Whole agent means it's the whole thing, right? It's the whole cell, the whole viral particle, whatever it is. So the MMR shot uses an attenuated whole agent vaccine. What is inactivated whole agent vaccine? Again, the whole agent thing tells you what? The whole thing is there. We didn't chop a piece of it off and put it in your body. We put in the whole bacterium. We put in the whole viral particle. But what did we do to it? What is inactivated a euphemism for? We killed it. Usually we heat killed it, okay? Now you have to be a little careful because if you're heat killing it, obviously you're going to denature the proteins, and some of those proteins might be antigenic. So you do this little dance. But you basically inactivate it in some way so that it's dead and not going to make you sick, but still has all of its spikes and all of its decorations that our immune system would recognize. So one of the polio vaccines uses an inactivated whole agent vaccine. Toxoid, something you would use, for instance, to vaccinate against tetanus. What is this? It's a toxin, and what did you do to the toxin to make sure it doesn't kill you? You basically inactivated it in some way. Okay, you did something to it that makes it not dangerous, but still retains enough of its original character to be recognized by the immune system, to be antigenic. A couple of newer forms of vaccines that have been really useful. So we have subunit vaccines. For instance, the acellular pertussis and one of the Hep B vaccines use this technology. What does this suggest? A subunit vaccine, is it a whole agent vaccine? Are you putting in the whole thing? What are you putting in? A piece, some subunit, right? It might be the capsule, or you might chop the bacteria up and extract the capsule and put the capsule in. Since this is safer in some ways. What is the benefit of using something like a subunit vaccine? There's really zero potential for for the original organism to multiply since you chopped it into pieces, right? Now, is it going to be good to do this with everything? Consider, are there pieces or things inside a once you killed off, say, a gram-negative, is it still dangerous? Could it still be dangerous? What's in it that would make you sick? That lipid in the lipopolysaccharide layer, that's an endotoxin, right? So you do have to be a little bit careful what you're using. But you can chop off something that isn't going to hurt you, but would still be, look, this is what the organism looks like, or at least what part of it looks like. Now, what is the downside of using a subunit vaccine? This might not be so obvious, but think about it. What might be a downside? Yes, several places in the room are coming up with the right answer. The whole organism has lots of different things on the surface that might be recognized by the immune system. So we're going to get a faster, more complete immune response with a whole agent. But that comes with some risk, right? If you didn't kill it, if you didn't weaken it enough, you could still make someone sick. This is going to be safer, but maybe not as effective depending on what you're doing. Nucleic acid or DNA vaccines. This has been used for to vaccinate horses against West Nile, and there's actually a few other applications for it now. So this is sort of the newest generation of viruses. What is a DNA vaccine? What do you think you're putting in to the? You're putting in DNA. Okay, so why would you do that? What's the point? What do you think is happening to that DNA? As crazy it's going to sound, what is the DNA? Usually it's injected into muscle tissue somewhere that the cells actually can get hold of it, and what are they going to do with that DNA? They're going to use it to make some protein, okay? Now, what is the point of doing that? Why is that helpful? What kind of protein would you put in on the DNA? What is it that your immune system is responding to when it sees an intact microbe, a virus or something like that? Are you talking about the interferons now? You are talking about interferons. And good for you for remembering interferons. That's awesome. We didn't talk about that in our little review, but no, that's not what's happening. That's a good guess, though. What is it that your body sees when a microbe comes into your body or a virus comes into your body? What is being detected? Antigens. And what kinds of things are antigenic? Proteins or sugars, right, that are sitting on the surface. So if you put in DNA and have your body make it into a protein, are you going to have it make protein that's normally found in your body? What are you going to have that DNA be for? Yeah, some kind of antigenic protein that the microbe you're trying to vaccinate against would normally make, right? Now, again, pluses and minuses. This turns out to be very effective against that particular thing that you're making. And it has a longer lasting effect than you might think, and it actually works, which you might not imagine it does, because the DNA is sort of out there, and how does it get into the cell, and how does the cell recognize and use it? That's kind of mysterious. But this is effective. Downside to this, same downside as for the subunit, which is what? It's just one thing. The whole microbe is a complicated organism with lots of epitopes. Kurt, you have a question. You're so patient.
Yeah. Why don't you put in proteins that DNA would use?
You could put in the proteins.
Is there reasons why you want the body to make the DNA versus the protein?
The DNA actually generates a much longer lasting, more effective response. So it's one way to think about it is you get more bang for your buck. So you put in one molecule of DNA, and how many proteins do you think will be made from that DNA? Lots, okay? Now you put in one protein, you just made one protein. So you get a much bigger response. Does that sort of make sense? You get a much bigger, and your body is making the protein, so it's going through that process, the protein showing up inside the body cell and then going up in the basket and being detected. All right. So if we want to make these vaccines, then sometimes it might be helpful to actually make some of the virus. Alvin. It depends on how you inactivated it and how effective that was. So for instance, a lot of times that means heat killing, so you just heat it for a certain period of time. And if you do it wrong, then yes, you could potentially still have some live organism in that mixture. Typically when people do this kind of technique, they have standards in place to test it and make sure that the batch is all good, but there is some low risk, absolutely. How do we make some viruses to make these vaccines? What do we do to culture viruses, and what is challenging about this? We need a host, right? Because unlike bacteria, we can't just slap it down on a plate with some food. We can't use just culture medium like TSB, which would allow us to grow lots of bacteria. If we put viral particles into TSB, what will happen? A whole lot of nothing will happen. Can we put viruses into cell culture? What is cell culture? Cell culture is a culture of cells, like bacterial cells or kidney cells or heart cells or whatever, brain cells. Can we do that? Can we put the virus in there? Now, do we have to be careful about what cells we're using? Why do we have to be careful? Because viruses are specific, so we have to use the right kind of host. We could use an intact host, so a whole bacterium, a whole plant, a whole animal, whatever the normal host is for that virus, or some reasonable facsimile. So there are some human viruses that can be grown in lab animals that are not the normal host, but they still will become infected if you do it right. Or, strangely, you can grow cultured viruses in embryonated eggs. So here is a picture of a chicken egg developing. There's the embryo, there's the yolk sac. And you can inject viruses into a variety of different places in this developing embryo. Into the egg sac, into the chorionic membrane, into the amniotic fluid, or into the amniotic cavity. There's a variety of different places you can inject viral particles, and they will actually grow. And so what that implies is that in those spaces, all the machinery that the virus needs, the ribosomes, the polymerase, whatever it is, the nucleic acids, the nucleotides, amino acids, all of the material that it needs, all the machinery it needs is there, right? Sometimes making vaccines is difficult for a variety of reasons, and one way to enhance the effectiveness of the vaccine is to use an adjuvant, so something that you add to the original vaccine that causes a more effective immune response. And so you can tweak this in a variety of ways. If you do that, whatever you've added, right, what do you have to be careful about when you add something into any medication that you're going to inject into somebody? A reaction, right? Whatever it is can't be toxic. And it also, you have to be careful not to put in something that on its own is immunogenic. And of course, we do know that some people have responses to different things, right? They may have an allergic response to the component of the vaccine that is not the actual thing we're trying to vaccinate that person against, but the adjuvant, the additive. So there's some risk, there's some obviously benefit to being vaccinated. We've talked about one of the risks is if you are adding stuff to the vaccine to get it into the person, you have to be careful about what that is. But what are some of the other risks depending on the type of vaccine that you're using? A whole Asian vaccine carries what kind of risk? You might make the person sick because what if you didn't kill it right, or what if it's weakened but not weakened enough? So, when we become vaccinated, what happens? That a vaccine contains something, right? Protein or some sugar, something that's antigenic, goes into our body, and then what happens? What does our immune system do to it? The B cell meets it, makes antibodies, talks to a T cell, all that stuff happens. Why do we need booster shots for some vaccines? Think about what it is that we're actually causing our bodies to do is we're simulating that primary exposure, which results in how is that secondary response saved? How do we know, what cells are we generating to allow that to happen? Memory cells. Do you think that those live forever? They do not. Okay, so one thing that boosters do is over time, and it could be a long time or it could be a relatively short time, those memory cells will die off, and boosters will help to make that, to sort of re-up your immunity. In younger children, it also helps to increase the pool of memory cells so that they will last much longer. Increase the pool of memory cells. Increase the population of memory cells that's in the body. So does exposure to every virus give us lifetime immunity? Why do we catch the cold over and over again? There's multiple strains. Not only that, but there's multiple different viruses that actually can give you the cold. And there's a couple different ones. But yes, it evolves fairly rapidly. There's multiple strains out there. Why do we have an annual flu shot? Why is the flu in particular so notorious? It has 7 chunks of its genome, which if 2 different ones get into, say, a pig, then they have a little field day and swap pieces, right? They have a little let's make a new flu day, and we suddenly have a new flu strain out there. Is that the only way we get new strains? No, right? Because there's also a certain level of just random mutation that happens. But the flu in particular is very rapidly evolving for that reason. Why is it that children and the elderly are at higher risk of becoming sick or contracting viruses and other kinds of pathogens in the first place? So children are potentially at a higher risk if they haven't been exposed to something. They also have a less mature immune system. The elderly, which we are all rapidly turning into, what is happening there? Our immune system's weaker, but what does that really mean? Why is that? Yeah, so all of the cells that are involved in this immune response are just aging and not as effective. Those memory cells, maybe that pool is getting smaller and smaller, and you did see chickenpox when you were, 5, but now you're 75, and so you may have reduced immunity over time for that reason, but also your whole immune response is not as effective. So vaccines are a really important application of all the stuff that we've learned about the immune system, and really did revolutionize medicine. There are a lot of other applications that are super cool. Some of them you'll recognize and be pretty easy to understand now what they're about. Some of them you'll recognize and be like, I had no idea that was how that worked. So there's a lot of diagnostic tests that we can do based on the fact that our bodies make antibodies, or antibodies exist, we can make them, and they have a very high specificity for their target. Yes, Kurt?
Do antibodies, are we creating them based on the antigen, or is it specific? Is the antibody coming from the antigen? So that's a really, that's a good question, and the answer is yes. So, and it's a subtlety that your book doesn't really go into very much, and I have not really emphasized, and I won't be testing you on. But so the sort of the initial thing is it's sort of the way you want to think about evolution, which is that it's random, right? So we have this whole suite of different variations on the antibodies. They're already existing. As B cells are being generated in our bodies, there's this region of the DNA in the B cell that's being shuffled around, and that creates all kinds of different prongs on the end of those antibodies, specific to a variety of hundreds of thousands of different possible things. Okay, those are already existing. They're just floating around, and as long as they haven't been killed off because they react against you, they are allowed to exist and float around looking for their antigen. If they find the antigen, then they proliferate. Now, one thing that happens is during that proliferation, more mutation can happen. And then they can evolve even more specificity and become more and more refined to that particular antigen, actually. So both things are happening, but it is fair to say that in the beginning, there's just a lot of random different guys looking for different kinds of antigens to start with. And if you randomly run into that guy, you're going to make an immune response against that. Does that answer your question? I thought I read something about, or I don't know how I came to this, but there's something to do with you build antibodies based on the antigen. Like you see whatever the outside protein is, and you build an antigen against that. Specifically, not just by mutation. Is that true?
No, so it does, but it does get refined. In the process of being activated, there is a process of further mutation. And that's going to randomly introduce variations in that whatever. Looking for a circle this size. Okay? Yeah. There is a way, so you might be thinking, there is a way that we can artificially generate antibodies against specifically what we're looking for, and we're going to talk about that. Okay, so antibody technology is being used a lot in diagnostics. One of the important questions is does the patient have disease X? But there are other things you could imagine, right? If you just think about human biology, which again is not the focus of this course,
there are a lot of things that your body might produce under certain stressful situations or in certain disease pathologies that are not microbial, which would be detected if we had the right antibody, or that you might be making antibodies against. So think about, for instance, hormones or cancer, different kinds of cancers. You might make really funky proteins that wouldn't show up otherwise, and we might have antibodies to detect those kinds of things. What we call biomarkers that show up in your body when you have a certain kind of disease that might not be microbial. They might not be contagious diseases caused by pathogens. They might be actually things that your body just does because it's aging or because it has cancer or something of that nature. So why are antibodies useful in detecting, for instance, if someone has a virus like HIV? How is antibody technology going to help us here? Say again?
They're specific to antigens.
Right.
The antibodies and it's being produced for some reason.
So you're exposed to some virus, let's say it's HIV. What does your body do about that? It tries to fight it, and one of the things it's going to do in the process of fighting it is make what? Antibodies. So then you could go looking for antibodies in the person's blood serum to see if there are antibodies specific against HIV. Now you know what HIV looks like, so you can put it in a dish. Then you can add the patient's serum and see, is there a reaction, right? That's sort of a simple... There are lots of variations on this theme that we're going to talk about. Oh, it's going to be so fun. A couple of things to think about when you're doing this kind of diagnostics with antibodies is sensitivity. So sensitivity is defined as the probability that the test is reactive if the specimen is a true positive. So let's think about that. If something is not sufficiently sensitive, you might have, 2 antibodies in your bloodstream, but that's too few to be detected. So this is not sensitive enough to tell you, yes, this person has seen HIV, but their antibody count is too low. It's below the level of sensitivity. Specificity is the probability that a positive will not be reactive if a specimen is a true negative. So specificity is you don't want the answer to come back yes if in fact the person doesn't have HIV. It's a false positive, right? So we have both of these things to consider. This sort of crudely was originally done when Koch was developing his whole systematic way of how do we figure out if this pathogen creates this disease. He injected guinea pigs with tuberculosis, right, which is caused by Mycobacterium tuberculosis. And the site of injection became red and swollen. So does this remind you of anything that you might have experienced? TB skin test. We still do it to this day. What are we actually looking for? We're checking to see if the body has what? Antibodies, right? Which would cause sort of a local inflammation.